Alright, hello Bio 245 yet again. So we have talked about excitation-contraction coupling. So the excitation of a neuron and the excitation of the muscle cell leading to contraction. But I didn't tell you how that contraction is happening, so that's where we're going to go. But first I want to talk a little bit about muscle morphology on the micro and the macro level. And from there we'll talk more about the actual contraction that's occurring. So first off, when you guys looked at the microscopes and we did tissues, you probably looked at the microscopes and you remember that we had those long ribbons with the red stripes or striations, right? So you might be wondering, how does what I saw in the microscope really fit into what we're going to be learning about with contraction and muscle? You might not be thinking that, but you should. So here's kind of what you should have seen when you looked at the microscope, right? That's our muscle tissue. And I'm going to talk a little bit about what we're actually seeing. So first off, let's think about the muscle as a whole. So whether that's your biceps brachii or your guns, as you can see, mine is excessive, so don't be too scared. But here's our muscle as a whole, okay? If you look at that muscle and you break it down, here's this little arrow. Whenever you see an arrow that broadens like that, that indicates zooming in or changing the angle or something. In this case, we're changing the angle of our muscle. We're doing a cross section so that we can look on the inside. So we're taking that muscle lengthwise and flipping it around so we can see it dead on. Okay, and we've cut it in half. When you cut open a muscle, you're going to see a couple different things. First off, you're going to see all these bundles going on. So you can see here I got lazy and just colored it red. But we have these big bundles when we look at our muscle. And these bundles are referred to as fascicles. Fascicles. So this whole thing here is the muscle. So we just took that muscle, flipped it around. And it's made up of these bundles that are referred to as fascicles. If you look at these fascicles and kind of dissect them out, you'll see that they are also made of smaller parts. So here's a fascicle that's been extended out. So if you can imagine that your muscle is a long tube, so everything that makes up your muscle will also be a long tube. So this is me dissecting out the fascicle as a long tube. Okay. So here's that fascicle coming out three-dimensionally. And you can see here that it's made up of smaller parts, these little smaller circles. If you take those circles, and again, we're made up of long tubes, so as we keep extending out, you're going to see more long tubes. But as we take out that fascicle, you'll see that it's actually made up of individual muscle fibers, which is synonymous with muscle cells. So we have our whole muscle made up of fascicles, and those fascicles are made up of muscle cells. You'll notice that I have a couple other words written up on here. You guys on your list should know that you are forced to know about the connective tissue layers that surround the muscle, surround the fascicles, and surround our individual muscle fibers. If you recall learning about locations and functions of connective tissue, I mentioned that for dense irregular connective tissue, that one of its functions is surrounding a muscle. Here you go. Okay, so we have that dense irregular connective tissue surrounding that muscle, keeping everything together, because that muscle is just made up of bundles, right? So we got to keep those bundles together in a thing. Same with our fascicles. Those individual fascicles are also surrounded by connective tissue, keeping the bundled up cells together, hugging them, keeping them close. Okay? And each individual cell is protected and coated with its own layer of connective tissue. And as you can imagine, these all have their own special names. One thing I want to do to tell you about these names is preface with the fact that whenever we're talking about muscle, we're either going to be using myo, so M-Y-O, that always means muscle, or we're going to just be using M-Y, so like in this case we have M-Y-S. Okay, so you always see MY somewhere when we're talking about muscle. So when we're talking about the layer of connective tissue that surrounds the entire muscle, we're talking about epimysium. So let me write that a little bigger so you can actually see. You should see it on your list. But we have epimysium. And you guys should remember for that epi always means on the outside of or on the ends of. For example, our epidermis for our integument, epidermis is the most outer layer. Or if we're talking about the bone, the epiphysis, the epiphysis, API, epi, always like that meant on the ends. So in this case, epimysium means on the outside of the muscle, MYS, okay? Next, we have the layer of connective tissue that surrounds each individual fascicle. That layer of connective tissue is called the perimysium. Perimysium. So peri is some kind of intermediate between outside and inside. Okay. So that's our perimysium. I'm just going to write peri so you can see. Okay. Lastly, we have that layer of connective tissue surrounding each and every individual muscle fiber. So you can see here it's colored in green for emphasis around each muscle fiber. And that is, surprise, the endomysium. Because remember, endo means the most inside. most inside, so endo, inside. So epi is the outside, peri is some kind of intermediate, endo is the most inside. So now that we know about all that and we've covered basic macro, the other thing I want to point out is here's our muscle fiber, right? Our muscle cell. You'll notice that it is striped red. Does that look a little bit familiar maybe? Like when you look at the microscope? Each of these guys is an individual muscle cell. Or muscle fiber, however you want to say. Muscle fiber is a term on your sheet, so you can stick with that, but just know that it's synonymous with muscle cell. Okay? So each one of these ribbons, each one of these long, long, long tubular ribbons is going to be a muscle cell. There you go, your muscle cells. If you pick apart that muscle cell further, the muscle cell is going to have the normal parts you would expect. It's got nuclei hanging out on there. Life is great. Remember, they are multinucleated. They're going to have all those mitochondria inside. If you remember talking about ATP and because mitochondria make ATP and talking about that, muscle cells need tons of ATP, so they got tons of mitochondria. They're going to have endoplasmic reticulum, which we're going to talk about. So all this stuff inherent to the muscle cell. Okay. But what it also has, you guys might remember on your sheet that we had to learn about cytoskeletons, right? Cytoskeleton, I told you guys, was just that network of proteins that gives the cell shape and also helps move stuff around. In the case of the muscle fiber, they are actually, they have a cytoskeleton, but that cytoskeleton is very compacted, very dense, and it fills the majority of the cell. And you'll see that when we cover the muscle fiber model, that most of that cell is the cytoskeleton made up of various proteins. And we'll talk about the specific proteins too. The proteins are arranged into something called a myofibril. A myofibril. So you have your muscle fiber or your muscle cell made up of these long tubular segments of compacted proteins called a myofibril. Okay? Now if you take that myofibril, again, this is just going from big to small. So if you take that myofibril, and again, here's one of those arrows indicating that we're zooming in. If you take that myofibril and you do and you cut along the edge of it, I guess do like a frontal section. I don't know if that really works. But in other words, if you took your myofibril and you cut it open so you could see inside of it, this is more or less what it would look like. You have these branches that are, I'll describe them. First off, we have these black lines, as you can see, and we have these red lines. These lines individually are referred to as myofilaments. And if we turn this just a little bit, you'll see that slightly better. There we go. So we have myofilaments, and the black filaments are called thin filaments, and the red ones are called thick filaments. Okay? So again, myofilaments. Myofilaments, you get a bunch of them, put them together, you get a myofibril. And that myofibril is just a tube full of tightly packed proteins that takes up the majority of the cellular space inside of the muscle cell fiber, or muscle fiber. The other things that you're going to see are how we got our stripes. So if you look at our muscle fiber, again, you'll see it's red and white stripes all the way across. What we're seeing is we see these red, thick filaments, and they look fatter, and they, when the light shines through them, they look red. And then you see light shining through the thin filaments, so it looks white. Since they're so thin, when the light shines through, it just passes right through. So wherever there's red or thick filaments, that's where it's the red stripe. Wherever it's thin filaments that are too thin to really block light, that's going to be your white stripes. The red stripes are referred to as the A band. A band. I always think it's darker, so dark has the letter A in it, so there's the A band. The white stripe where you see only thin filaments, that's the I band. And I always think light or white has the letter I in it, so that's I band. Okay? The other thing that you gotta know is, see how there's this central line going through the thin filaments that kind of acts as a supportive structure so the thin filaments can stick out of the side? So here's a thin filament, here's a thin filament, here's a filament, here's that supportive structure. That supportive structure is referred to as the Z-line or Z-disc. You'll see it called different things in different books. I'm going to call it the Z-line. You might hear me say it's a Z-disc every now and then. But Z-line, so that's going to be our supportive scaffold for our thin filaments to stick off of. The other important thing that you got to know is sarcomere. That's the other prefix. So I told you guys whenever you see myo or mes, M-Y-S, you should think muscle. Same thing with sarco. If you ever see sarco, that means muscle as well. Okay? So from this Z line to this Z line, we have a sarcomere. Sarcomere. Okay, so from Z line to Z line is a sarcomere. The reason why we care about sarcomere is because we have these different Z lines on the end, and when the muscle finally contracts, the Z lines move closer together, which results in the shortening of the myofibril, shortening of the muscle fiber, and ultimately shortening of that fascicle, and therefore shortening of the muscle as a whole, because that's how a muscle does its job, right? A muscle doesn't just lay there like a limp noodle. It actually physically shortens and pulls. It acts like a pulley, okay? So whenever the sarcomere is active and it's doing contracting stuff, it will physically become shorter, and we'll talk about how that happens. But now that I've introduced you to all of these terms, I'm just going to do a real quick recap. Again, when you're looking in your microscope, what you're actually seeing are individual muscle cells and muscle fibers. So how does that pan out overall? Again, here's our muscle. If you zoom in, if you do a cross-section, you'll see that we have fascicles, and that inside of those fascicles, we have a bunch of muscle cells. So here's our muscle cells being dissected out. Inside of that muscle cell, we have normal cell stuff, plus we have this compacted bundle of proteins referred to as a myofibril. And this is where all that contraction is actually going to be occurring on the ground level. If you take apart those myofibrils, it's made up of individual myofilaments. Myofilaments. Those myofilaments come in, two flavors. Let's move this over so you can actually see. Those myofilaments come in two flavors. Thick, which are red, and thin, which are just black or really thin. Light passes right through. When you look at the actual composition of the myofibril and how these protein fibers are arranged, we have our A bands, which is where you have your thick filaments, and you have your I bands where you only see your thin filaments. We have a sarcomere, which goes from Z line to Z line, and the Z line is just that scaffolding supportive structure for our thin filaments. So come back to part 2 so we can actually talk about why this stuff matters and how they work together to result in contraction.